Hi everyone, welcome to the channel. Today we are going to cover study guide for lab test 2 at bsc1010 lab. We have 3 labs actually covered in this review. They are membranes, enzymes, and photosynthesis. Okay, again, this is a study guide, just a brief review for every test, like as in other tests. Please make sure that you complete all the connect labs and then review your labs and get your data, review them, and then study these specific 3 labs. So let's go ahead and get started. So first, the membranes. So in membranes lab, we have diffusion, right, from high concentration to the low concentration. Basically osmosis, which is diffusion of water across a membrane, like plasma membrane. We have salt solutions, and we can have 3 different situations, like hypertonic if it is high salt outside, hypotonic if it is high inside, and then lastly isotonic when they reach same level. So for plants, that's called flaccid. For animal cells, it's called normal. For plants, when it's hypertonic outside, they become plasmolyzed and they lose water. And also there is diffusion in semi-solids, like in agar, and how chemicals move from one place to another place on agar. Make sure that you know how to find the isotonic point, and then make sure you can compare and contrast. For this lab and next lab, we have serial dilation. So there is a formula called C1 times V1 equals C2 times V2. So you can actually plug in that and then find out your serial dilations. So they could be 2-fold, 10-fold. So always the last tube is your control or negative control with 0 concentration. Don't forget. Next lab is enzymes. So enzymes lab, we have enzyme and substrate. Every enzyme attached to a substrate, they need a substrate for their active site, perfectly fits to their active site. And when they do that, they become ES, or enzyme substrate combo. And then the next is they produce a product, and then enzymes leaves intact and then moves on to the next substrate. Enzymes are very sensitive to temperature, pH, concentrations, so they must have optimal pH concentration and pH and up to temperature. So make sure that you know what's the enzyme name, what's the substrate name, what's the optimal temperature, pH, and concentrations that we actually did experiment on and find out about our enzyme of the choice for this lab. Again, we use serial dilation to find the concentration that's optimal concentration, the best concentration for this enzyme work in that specific substrate. The next step is photosynthesis. Basically, it's about the most important reaction on biosphere: photosynthesis, pigments, light, chlorophyll, CO2 uptake, light versus dark in different light wavelengths, right? So remember, photosynthesis has light reactions and Calvin cycle. So remember the formula for in order to start the photosynthesis, we must have water, we must have light, we must have chlorophyll, and then CO2. The products are oxygen, sugars, and water again. So we use a number of plants in photosynthesis lab. And so make sure that you be familiar with the plant names and then what experiments we're using them and which techniques, how do we measure the light absorption. So make sure that you be familiar with that. And also how do we separate the pigments. A pigment is always, remember, a colorful molecule in plants that could be giving the plants those colors you see, like orange and green and red and yellow, etc. And then make sure that you know which plant we use in which experiment. So just review these together with your Connect Labs, and good luck in your testing next week. Thanks for watching, and please feel free to comment, subscribe, and see you next time.